Hi, Michelle Glass here, and welcome back to another episode in our Chapter 28, Lecture 28 series. And the topic for this video is spermatogenesis, which is the formation of sperm cells. This is beginning for males with the onset of puberty, and it will continue until, late in life. So this is very different from females. We'll see there's a specific window when gametes are produced in the female life cycle. The formation of gametes, in this case sperm cells, follows the process of meiosis. I'm going to talk about it real briefly in this video, so if you're not super strong on that concept, make sure you check out my other video in the playlist or do some research to make sure that you're okay with that topic. And then the last thing, we've already talked about sperm production is happening inside the nurse cells, which are found in the seminiferous tubules. So of course, we are in the male gonad, which is the testis for this topic here. When we're talking about spermatogenesis, the formation of any gamete, it's going to involve meiosis. So remember, if you're not super strong on this topic, I have an entire video dedicated to meiosis, and I've added it to our playlist here. In meiosis, you start with a cell that is diploid. Diploid, remember, means a complete set of chromosomes. So in the case of humans, the diploid number is 46. Now, before meiosis even occurs, the DNA is replicated, so you're going to actually have 92 chromatids. So that lets you go through your first meiotic division. Meiosis is very similar to mitosis in that you're going to have a prophase, metaphase, anaphase, telophase. Remember, with prophase, the DNA is going to condense into chromosomes, the nuclear envelope disappears, and the spindle apparatus forms. And metaphase, they line up in the middle of the cell. And anaphase, they pull away from each other. And in telophase, you get two new nuclei. There's a few important differences in meiosis 1. Key things are happening in prophase 1. You're going to get an event called crossing over. And this is where you're going to switch pieces of DNA that are in a homologous pair. So you're going to switch DNA between non-sister chromatids of a homologous pair. Non-sister chromatids of a homologous pair. So you get a new combination of genetic information showing up. And then in metaphase 1, you're going to see that the homologous chromosomes line up in pairs in the middle. So that's new. At the end of meiosis 1, we get a cell division, and so now we have our homologous chromosomes separating into our two new cells. They go through a second division, so you have a prophase 2, metaphase 2, anaphase 2, and telophase 2, resulting in 4, count them, 4 haploid daughter cells. Haploid, remember, means half of the genetic information, so we will see 23 chromosomes in our sperm cells. Because of crossover event and random orientation of those homologous pairs, we will see that all 4 of these chromatids, all 4 of these daughter cells will be very different from each other. Okay. In the sperm production, you start out with a stem cell called a spermatogonia. So the spermatogonia is the stem cell. It's a diploid cell, and it goes through mitosis, where it makes 2 daughter cells. So mitosis, remember, is more of the same. So it's going to make a replacement for itself. It's going to keep a spermatogonia that's there available to do this again in the next cycle. And then it's also going to produce a cell that is called the primary spermatocyte. The primary spermatocyte is the cell that's going to go through meiosis. So the primary spermatocyte goes through meiosis, producing secondary spermatocytes. Each one of these secondary spermatocytes is going to go through a second meiotic division, which is going to result in the formation of 4 unique haploid spermatids. So at the end of meiosis 2, we get 4 spermatids, which will ultimately become the sperm cells. Just as a couple of quick reminders, all of this is taking place inside of the nurse cell, and the nurse cell is located in those seminiferous tubules, so we're in the testis. And the starting out primary spermatocyte is a diploid cell, goes through meiosis 1, homologous chromosomes separate. You get secondary spermatocytes, which go through meiosis 2. And during this division, sister chromatids split. So now we have 4 haploid spermatids. And because crossover occurred during prophase 1, each one of these cells is unique. Now, the spermatid is going to go through a maturation process still inside the nurse cell, and we're going to call this process spermiogenesis. Spermiogenesis is the maturation of the spermatid to the sperm. After spermiogenesis has occurred, then we're going to see spermiation happening. Spermiation is when the sperm cell leaves the nurse cell and it enters the lumen or the cavity of the seminiferous tubule. So now it's getting ready to head on to the rete testis and the efferent ductules and onto the epididymis. Now, the sperm cell is going to spend about 3 weeks in the epididymis where it's going to do its final maturation. And then it's not going to become motile until it enters the ejaculatory duct and mixes with the seminiferous gland secretions, seminal gland secretions. I used the wrong term there. Seminal gland secretions. And then it's still not capable of actually fertilizing an egg cell until it has been exposed to enzymes and conditions inside the female reproductive tract. These last 2 steps of becoming motile and capable of fertilization is what's called capacitation. So even though you have a mature sperm cell leaving the epididymis, they're not fully ready to fertilize an egg until after they've become motile with the seminal gland secretions, and then they have to actually go into the female reproductive tract in order to be then capable of fertilization. Taking a look at a mature sperm cell, which can also be referred to as spermatozoan, that would be the singular version. Spermatozoa would be plural. We see that it has a head and a neck and a tail. In the head, that's basically where you're going to house the nucleus. Remember, the nucleus is haploid, so it has 23 chromosomes. It's going to have at the very tippy top what's called an acrosome. These are going to be the enzymes that are going to be important for fertilization. Fertilization requires that the sperm cell actually penetrates the egg and gets those nuclei to fuse. In the neck region, you're going to see a big bunch of mitochondria. These are going to be providing the ATP that's required to propel that flagella. I shouldn't say propel the flagella, but to power the flagella, which will then propel the cell as it's moving through the male urethra and then the female reproductive tract. We'll see that it has to make its way all the way up into the uterine tube of the female in order to reach an egg. So it has quite some distance to travel for a little microscopic cell. Essentially, the sperm cell has, as part of that maturation process, it's lost a lot of the organelles. So that is basically just this little tiny packet of chromosomes with the machinery necessary to actually propel it to that egg. All right, so that's it for now.